And with that, I will have to move on because I think we're already running, well, not super late, but I do want you to have some time to explore your own data or to continue with me on the other data set that we have. And maybe I will quickly ask if there are any questions so far. Otherwise, or maybe I will first explain what we're going to do next and then ask for questions.
There's one question in the chat right now. Oh, maybe now. Yeah. Sorry, Eric answered it.
Okay. Yeah, I think I'll just quickly tell you what the plan is for the next hour, and then you can ask some questions because you might have also questions about that. So what are we going to do next? So now I want you to work either on your own data or on a second example data set. The second example data set has been sent to you either via email or you have downloaded it from the OwnCloud. Both of them, the one in the email and from the OwnCloud, show the same colony. It's just that one of them is only 2-dimensional, the other one is 3-dimensional. If you want to follow along and your computer is not very powerful, I recommend doing the 2-dimensional one because it just moves much faster.
May I ask you one question? In terms of the segmentation, this is then always the cube-based one, effectively, or does BiofilmQ ever try to, do a watershed segmentation or so? I mean, when you have the cell count.
So we have not incorporated this. I think we are thinking sometimes about this, but since the deep learning is so ahead, kind of, we are more supporting or thinking about or supporting that a really good segmentation is done via deep learning, a separate framework, and then the data is imported, and then we can calculate on the segmentation that is imported the properties. So there's I think this is like a branch we are going into, that the very good segmentations need to come from outside, from other tools, because they are just better. And BiofilmQ is mainly, or can only support classical stuff, and classical segmentations are just not good enough anymore.
Okay. But when you do segment that with deep learning or anything, then I'm still trying to understand how that relates then to the cube. So if we have, for example, cell number as a parameter, is that then the number of segmented cells, or is that still cube based?
No. So if you import segmentation results from a deep learning or from another segmentation algorithm, we don't need the cubes anymore. The cubes are something like actually they are an alternative to a watershed. So we have this threshold, and because cells are clumped together so much that sometimes there's it's not so simple to use a watershed because it just doesn't work good. We just cut it in cubes. So we just say every 10 pixels we have a cube, and after 10 pixels there's the next cube. So we create these pseudo cells that have no resemblance directly to where a real cell is, just by thresholding. And because watershed is it's not working good in
so but then so basically the cell number as a parameter, if I'm going to go for larger cubes, I will have a lower cell number, so it's just an approximation.
Yes, in the cube method, yes.
Okay, and I wasn't sure because you have that combined graph where you have the segmented bacteria growing, and then you have the cubes inside of that. So I wasn't sure if there is a combination of cubes with, for example, pre-existing AI segmentation or deep learning segmentation. So does it have to do things differently if you have a deep learning based segmentation that you import? What do you actually import there?
Label maps, or do you know label maps?
Say again, label.
So this means it's or label image. It's an image. It's an image with the same amount of pixels as the image, as the grayscale image, and for the background is 0, so everything is 0. And for each instance that is a cell, you get a unique number. So then it's labeled from 1, 2, 3, 4, 5 for each separate cell, and therefore cells that collide, they have different numbers and therefore can be separated. And this is the thing that needs to get into BiofilmQ. And from this, BiofilmQ can calculate everything again.
Okay.
So if I understand correct, and Hannah, correct me, please. The cube segmentation method is just creating something like this label map, just on the basis of cubes and the thresholding.
Yeah.
Whereas,
okay.
Yeah, that is true. And actually, now I'm just quickly sharing here. So this is a slide that would have come up in an hour roughly. So this is what these label maps look like, or these label images. So this is just one slice, and the intensity values correspond to kind of a cell ID. And this is something that you import, and then you can use your neural network-based segmentation, and basically instead of using cubes as objects and calculating everything for cubes, it's going to use your cells as objects and calculate everything for cells. So it's true, like Niklas said, just the cubes are basically also the same kind of structure in MATLAB. So if you ever work with MATLAB, MATLAB has this thing called a structure, and there is a certain type of structure that kind of relates to an image in which objects are present, and each object has a different ID. And just like every cell has a different ID, every cube has a different ID. So internally, it's actually not a big difference between cubes and cells.
Okay. Great, thanks. And one other question: are you ever considering putting in tracking as well? I mean, so because I will be also attending the Mastodon workshop, and I could just see a lot of this working together, apart from of course them being different languages.
So the tracking is a little bit similar to the segmentation. We know that there are better tracking algorithms out there. We have like a very simple tracking, which is just an overlap-based tracking, which will work if you have really high time resolution. But of course, it doesn't really like there's some such better things out there, and we always feel like our algorithms are basically not powerful enough, and then the tracking can or may be done in a different tool. But so we have this cube tracking, which is it's not really a tracking. It's more like a how a biofilm grows, like a little bit of a lineage thing. But I think what you're looking for is a different type of tracking, probably a single cell tracking, right? Like moving cells maybe, or cell lineages. Yeah. And I think in the current implementation of BiofilmQ, this is not really sophisticated.
Okay. Yeah, but well, thank you very much. I think it looks incredibly powerful. I had to iron out a stupid mistake at the beginning, but Eric helped me with that. And yeah, no, it looks very good, and I'm looking forward to use it more.
Thanks a lot.
Thanks.
Thank you very much. Yeah.
Okay, I think now we're going to continue. So I'm going to share my screen again.
Yeah.
Yeah. Sorry.
Can you elaborate on the question of Jarel with the version number?
I didn't see. Is it in the
it's
chat?
In the chat. He asks about the MATLAB version of 2015 B, and
is it? have you tried if it's working with 2050? Because I think this is a version where it should be working.
Yeah, he says it works, but some parts that do not work.
Okay. So
we need
so
to
I think this is like a specific thing we would have to look into, why it's not, or what parts are not working. I think this might be something that maybe if you have a specific problem in the plotting, you could contact us in the forum, for example, or via email, and then we can have a look specifically at why this is happening and maybe find a fix for it.
Yeah.
But I'm guessing that there are maybe just like some, some commands that are not in this version of MATLAB that appeared later in a MATLAB version that need to be replaced. It's a little bit difficult to answer that without knowing what exactly is not working. Okay, but yeah, so be sure to contact us about that. So yeah, that's also something that Philip just mentioned. Of course, not everything happens from the start, and BiofilmQ is still like a work in progress. I mean, this is like, we are some PhD students, like trying to get a good software out there, but obviously it has its flaws, and we're very happy and very thankful for people telling us when things are not going as they should, and we're always also happy to help getting it running for you. So I understand that some things can still be improved, and we're always happy to hear about those things. So this is just something that I will probably also come back to in the end, but just because we just had it coming up here. All right, so I will continue with this here with this example data, and you can probably see this file here, and I'm not sure if you've been able to open the file that I sent via email yet, or if not, that's also fine. So what I want to show you with this example is just like something that is completely different from what we've seen before. So before we had a biofilm in high resolution that was like 100x with 2x lens, so like 200 times the magnification. We could see single cells. So this was really very different from what we see here. What we see here is we have a colony growing on an agar surface. It was spotted kind of here in the center where they're still like not really, where you can't see the lines yet. And then as they grow outwards, I think you have seen these sort of competition experiments before, possibly. If not, you can probably Google these images. I think are really, really fascinating to look at. So the idea is that you have two strains growing together, and you want to know if eventually one strain kind of outcompetes the other strain. So if, for example, you would see that over time, everything was turning red. And when I say over time, I'm talking about from inwards to outwards. Everything was turning red, it would mean that like the red strain outcompetes the green one. And now I have already mentioned something that is very important about this colony. I've been talking about time and spatial resolution, but in fact, in this case, we only need the spatial resolution because the space is also beautifully capturing time, because as the cells are growing outwards, so they start out in the center of the plate and then they're growing outwards, they grow outwards continuously. Therefore, this is sort of our time axis. So as we are going to explore this colony, we are going to refer to like time or like over time as from inside to the outside of the colony. And this is, of course, again, the spatial measure of distance to the biofilm center or distance to the biofilm surface, as you wish to define it. And this is something that we can calculate in BiofilmQ. And what we see in this image is, again, these two strains growing together. You can see also there is some competition happening here. For example, we have a green lineage that kind of dies out and is just overtaken by red. And it's just very interesting picture. And of course, also there is no scale bar here, but this is like a millimeter size. So I think maybe the whole thing is a centimeter or two in diameter. So this is a large colony. We will not be able to have any single cell resolution in there unless you have a gigantic mesolens setup. So what we're going to look at is we want to look at how these strains grow together over time. We want to know how the abundance of the strains over time has changed. And the way that we do this is, like I said, over time, to us means distance to the biofilm center. And the abundance is something that we can measure in BiofilmQ. So in the end, the plot that we want to get out will look somewhat like this one here. where we have the relative abundance of channel 1 or cells fluorescent in channel 1, so which would be the red one in this case, over the distance to the biome center. And I want to show you this workflow here, which consists of first importing the data. This is something that you will have to do almost all the time with your own data as well. So this is something that is also good to have seen once. Then we're going to segment both of these channels. Remember that before we only segmented one of the channels. We're going to merge them. I have mentioned the merging in the first part of this workshop. However, now we're actually going to do it, and then we're going to calculate this distance and visualize. So this is roughly the workflow that we're going through right now, starting with the import. And now I'm going to switch back to BiofilmQ. And in fact, what I'm going to do is I'm going to close BiofilmQ and reopen it. Why am I doing this? It's just cleaner, and I would recommend that if you switch from one type of image analysis to another one, you also close your BiofilmQ window and reopen it, just so that all of the settings are basically refreshed. Because what happens if you open a directory in a BiofilmQ that has already been kind of like opened, the parameters are copied, because BiofilmQ assumes that basically when you work in the same BiofilmQ window, all of the different folders that you open will be of the same type of analysis, which usually makes sense, because usually what you're doing is you're doing the same type of analysis, just on different folders. So maybe just have two MATLABs open if you want to use it on different types of images, on different types of analyses, or just open a new MATLAB BiofilmQ window. So this is just you can do that right now at home if you're following, but it's not crucial. It's just for convenience. And now the files that we are going to look at are located in here. So we have two files in here. One is a 2D file, one is a 3D file. I have just placed them both in here. You can choose the 2D file if you're following along at home, just to speed things up a little bit. And also to see, I guess, that a 2D file actually works with BiofilmQ, because BiofilmQ is optimized for three-dimensional processing, but two-dimensional processing is possible. And we would like you, you can also try it here. All right, so I'm going to browse, and I'm going to move to this folder here. And you can see when I open this now that the view looks different from what we had before. So you might remember that in here we had an image. This has disappeared, but well, but more importantly, we don't have the sequence of different things that we can do here on the right side. We only have one tab, which is called importative. Because as you have these ND2 files, you can actually only use them in BiofilmQ if you have imported them. What we see in the center is, instead of an image, we see some information about the image that is in this file. We see that this is a file which contains only one position. And in this position, there is one image consisting of X and Y 1024 by 1024 and 5 layers. We have 2 channels and 1 time point. If I look at this one here, it's very similar, only that it only has 1 layer. So this is really the same colony, just like several layers. And if I want to import files, I can again choose them in my image range. I'm just going to import both of them right now, because I think it is okay in the interest of time. And what you can choose here, you basically only have one option that you can choose from. You can decide if you want to have one experiment folder per position during the import. Now, in this case, it won't make a difference, because I only have one position, and therefore it doesn't matter if I generate like several folders per position, because I only have one. If you have several positions and you have a time series, I would always recommend creating several folders during the import, because otherwise you will have a mixture of different time points and different positions in the same directory, and BiofilmQ can't really handle that. Only if you have different positions but only one time point, then you can like not check this box and keep it all in one directory. So for now, since it doesn't matter, we will just keep this unchecked, and we will click on import files, which will convert the files to the type that we need. And you see this was very fast, also because the files are not very large. And I am informed that there are two subdirectories which have been created. And I can choose if I want to switch to the first one. I'm just going to do that. So BiofilmQ then automatically load the first one of them. Just to show you what has happened in this folder here. So originally we only had these two files. We now have two additional MATLAB files, which BiofilmQ automatically generates when it opens the directory. But more importantly, we have two folders. And these are the folders that BiofilmQ has created, and then BiofilmQ has placed the files in there. So if we look at this example colony, I do find the TIFF file again that I always have. Again, this is a TIFF file with 6 layers. The bottom one is an overview plane, and the other one is the 5 layers of the image. I have this for channel 1 as well as channel 2, and I have a metadata file for both channels as well. So the import has worked. We are now in this view again, so now we can start working with the data. We can check what it looks like. So this is our image. We can see we have these two channels, and now what we want to do is we want to segment them. I think I'm going to go relatively quickly to these through these steps today on the second part, because we've already talked in more detail about it in the first part. I'm going to go straight to the denoising, and I'm going to uncheck the top hat filter. Why do I want to uncheck the top hat filter? Because the patterns that I can see in here are of different sizes. Well, first of all, I don't have single cells, and the top hat filter is optimized to better resolve single cells during segmentation. But also these patterns that I see in here have different sizes, and therefore it wouldn't really make sense to apply a top hat filter with the same size to the whole image, because you would basically have to adapt it to these pattern sizes. And in general, it doesn't really make sense to apply a top hat filter to an image that doesn't have single cells in them or anything that resembles single cells. So here we'll not use the top hat filter for now. We'll keep the denoising as it is and go straight to the thresholding. I think we're going to use also thresholding here, and switch again to after image processing just to have a look at what also thresholding produces in this case. And we can see here the colony is detected, but it seems as if we have a little bit too much cut off here at the bottom. And we are going to correct that by decreasing this sensitivity value. So I'm going to decrease it a little bit. Let's maybe put it 0.65 or so. So I want to put like a, yeah, that looks fine. So here, okay, so in the bottom layer, I'm still missing some of the colony, but in the main layer, like in the central layers, the colony is kind of well defined. So I'm going to pick that, and I'm going to check the clumping part. Now, what is this, the CubeSat length that we should choose here? And this is again a case where we need to kind of like think about what is the type of, what is the pattern that we want to resolve, and what is the order of magnitude in space, or in what is the scale of these patterns. And here I think the smallest pattern that we want to resolve is these like very fine structures that go to the end. We don't really know yet how large they are, so I'm just going to pick some cube size length that I think makes sense. We will later then look at it if it made sense or not. In the post-processing, I'm just going to remove small areas just to make sure that we don't have any dirt or any noise. And then I'm just going to click on segment. So now the image is segmenting. You can see this was relatively fast. We had 14 seconds, and I can have a look at the result. And this is now where I want to decide if my cube size makes sense or not. So I'm going to open this auto view, and here I've chosen the intensity mean channel 1. I hope this is not too fast for you. Otherwise, like if you follow along, so basically you select the image, you would open this segmentation preview here, and then select intensity mean channel 1 and the auto view. And this is what it looks like. What we can see is we can see these very fine structures there and here. And we can see that the cubes are actually still quite a bit larger. And this is why I think we should decrease the cube size. So we're going to go back to the segmentation and think about what is a cube size that makes sense. And here I want to mention or point out that the cube size, of course, we want to have it as small as necessary, but we do need to keep in mind that the time for processing greatly increases as we decrease the cube size length. And I typically do not recommend for anyone to use less than 3 pixels or voxels as a cube side length if you have like a normally sized image like this one, for example, 1024 by 1024, just because it gets really, really slow. So in our case, I want to decrease it. I think I will decrease it to 4 so that we don't go too low, but we do go significantly lower. And now I'm going to segment this again with a cube size of 4 and see what the difference is like. And this is a little bit the downside of having now a smaller cube size. You can already see it takes longer. We need to wait for it a little bit longer. Let's have a look at the console. We will soon be informed how much longer it took. So I think we had 14 seconds before. Now it took us 40 seconds. So this is more than twice as long. So really this does have an impact. As I want to look at the segmentation preview now, I quickly need to switch between channels because so here this is something that I've actually mentioned before. It's a little bit of a bug that we're going to fix. The segmentation preview doesn't refresh after you resegment the cells, so you actually have to do something like refresh the folder, for example, in order to reload it, or you have to switch channels. Actually, refreshing the folder doesn't help, or you have to move in here. So what I'm going to do is I'm going to switch channels quickly so that it resets. And then I'm going to go back to the first channel. And here you can see the pattern of the second channel. So now that we're back in the first channel, I'm going to load the segmentation results again and have a look at the same property that we looked at before, which is this intensity mean channel 1. So select it, click on auto view to get this view that we had before. And as you can see now, the cubes are significantly smaller. And I think you can also see that they're now somewhat in the range. I don't think I can really zoom in here. But they're somewhat in the range of these fine structures that we want to resolve. So this is definitely much better than what we had before. And we're going to stick with this choice now. So now we're going to segment the second channel, and we want to do this the same way as we did for the first channel, which means we want to apply the same filters. We want to apply the same cube length, which is important if you want to do the merging later. Do, however, want to double check that the threshold works, because as the second channel might have a little bit of a different fluorescence profile, the thresholding might not be optimal anymore. So let's have a look at that. Kind of looks okay. However, I would go a little bit lower again with the maybe 0.4, so that we get all of these fine structures and they're actually resolved until the end. Maybe 0.35. I hope that it doesn't seem like it brings a lot of noise, so I think this is okay. So we'll pick this threshold, and then we will just click on segment cells, because everything else we want to keep the same. Okay, looks like the segmentation has finished, which means that we can now actually still have a waiting sign. Just look like it's finished. I don't really know why. Oh no, okay, I was too impatient. All right, so now it has finished, and we can do the merge. So why do we want to do a merging, and what does it actually mean to merge? The general idea is that if we want to look at the colony as a whole, which would be all the fluorescent part here, no matter which color, we cannot do that by only looking at one fluorescence or the other fluorescence. So if we look at the segmentation of channel 1, or we look at segmentation of channel 2, neither of them is capable of capturing the whole colony. If we, however, take them together, we will be able to calculate properties for the whole colony. And importantly, we will also generate information about the abundance. And now what does this abundance mean? If you think of a cube, and you think of a cube which is, for example, partly filled with cells that are fluorescent in the red channel and cells which are fluorescent in the green channel. And maybe it's filled half-half, might be filled 75 to 20, might also be that 20% of the cube is filled with one color and 20% with the other color, and the last 60% is kind of empty, there are no cells at all. So every cube will have some form of ratio between these two different colors in the cube. And this ratio is translates to an abundance in the cube that tells us how the two strains kind of compete with each other. And of course, we have chosen the cubes to be relatively small, which is why most of the cubes will be filled only with red fluorescent cells or only with green fluorescent cells. There will be some at the corner that are filled partly with red, partly with green. But overall, we will get the 0 or 100%. And then what comes into play is averaging over the distance to the biofilm center. So we average kind of over a ring in this colony. And then in this ring, in this colony, we average over all cubes and check how much red and how much green do we have in there. So I'm going to merge the channels now. The way that I do this is I choose channel 1 here in order to merge to channel 1. So this channel here, which is written in here, has to be the same as the channel in here. Now I decided that I want to merge channel 1, so I'm going to merge channel 2 to channel 1, and then I basically just press merge. And this is going to apply the merging. What it's doing in the background is it's loading both of the segmentation and it's putting them together. And as I do that, and now it asks me if I actually want to do that, also informs me that the original files will be copied to this folder, and we will look at this folder in a second. So what it does in the background, it combines the segmentation and then saves the new result. And as the merging is happening, I want to have a look with you at this data and actually also at this data folder. We haven't actually had a look at this data folder before. So this folder hasn't existed before, right? When we opened this file, when we opened this folder, we only had images and metadata files. Now this data folder has appeared, and that means a segmentation has been performed. If I look into this data folder, I can see that I have two files in here. The files have the same names as the files in here, except that they have this underscore data at the end. Sorry. And you can see that we have now also a new folder in here. And that is the one that was just created by the merge. What happens during the merging is that we will replace the segmentation of channel 1 with a merged segmentation. In order to be able to change it back to have like a backup, we copy the original segmentations to this non-merged data folder. This is just really see it as a backup in case you find that the merging didn't work as you expected. You can delete these two files here and put these two back, and that will undo the merge. So these data files, they represent the segmentation, everything you have calculated. Do not delete these data files unless you know what you're doing, because otherwise you will have to do all the segmentation and all the parameter calculation again. But also if you like MATLAB, and I think I'm going to do this. Do I have it? Yes, no. Okay. So what I can do actually is also look at these data files in MATLAB, because it's a MAT file. And I think it's worth having a short look at them just to show you what is in there. So I'm going to load it, and I'm going to load it to something called object. So this is really just for people who would like to potentially look at them in MATLAB. If you don't have MATLAB, if you don't like programming, then you can just like take a 2-second break while I talk, essentially. But the idea is that if I load these files, if I load this file, it will contain all the relevant information about the segmentation. And it takes a moment to load because it's actually not that small. And right now, as I loaded it, it tells me actually what is in there. We have a structure, which is like a MATLAB file type. It tells me how many objects I have in here. So currently I have 260,000 cubes. It tells me the image size, and we see it's 1024 by 1024 by 30. This is not 5. We had 5 was our set slices. It's 30 now because MATLAB or BiofilmQ interpolates the slices so that, remember, the spacing, the scaling is equal in all 3 dimensions after BiofilmQ has done this interpolation. That is why we have 30 here. And then it has some other things. It has, for example, params. It has global measurements, measurement fields, and it has stats. I quickly want to show you what stats looks like, because that will be the most relevant for you. Stats is the table that collects all information that we have about the data. This table contains fields. Fields are the names of the properties that we measure, and you can see here we have relative abundance properties. As predicted, those are typically either 0 or 100. But we also have things like volume, volume fraction, surface of the cube. So this is like a centroid. This is all the information we have about the cells, and now the rows correspond to the different cells. So here, for example, we have so many different objects that, of course, we can't go through them. But it is in principle good to know that this table exists, and this is exactly the table that would be exported into an Excel sheet if you do the export into an Excel sheet. So this is the stats part. And then the other part, and this is, I guess, well, the metadata might also be important. This, for example, contains if you have applied a cropping. We have not applied a cropping here, but if we had applied a cropping, it would have the range in here. So that might also be if you accidentally undo your cropping, you can find it in here. And there are some other informations, for example, also in the params that I will not go into detail. Just in very short, the params kind of collect all information, all the settings of every single checkbox, every single text field that you have. And it allows you to recreate the same settings again if you ever lose them. So this object file, this file that is saved as a segmentation file, is actually interesting and useful, and it's good to know that it exists. Even if you're not going to use it, it's still good to know that it exists. So the merge has completed. Let's have a look at the results. We will load the segmentation results from the merging. You can see now that this actually takes a moment. Remember that when I did this in the first part, it would almost immediately pop up. Now actually the file has to load because we have these, what was it, 160,000? 260,000 different cells or objects. And now we can see, okay, so this is my preview. If I look, for example, at the volume of a cube in the auto view, you can see now that the cubes that I have represent the entire colony. And now, if I was a little bit more of a perfectionist, I would probably say, oh, I'm not happy about these things sticking out. And this is, you can see here, probably I didn't do the thresholding perfectly. And this is why in some places, for channel 2 specifically, channel 2 has like the threshold is a little bit too low. That's why we have this sticking out here. But, you know, for the purpose of today, I'm not very much of a perfectionist. I'm happy as it is. I'm happy that the whole colony is filled with cubes, at least in the central layers. And now, what information do these cubes contain? So the merging creates these 3 different fields. Let's start with the overlap. The overlap tells us in the cube what part of the red and the green channel overlapped. So if this is yellow, and now we can look at the color map, this is between 0 and 100. If this is yellow, it means that due to our segmentation, the cube was filled completely with red and completely with green. And we can see in which areas this is the case. For example, take this area here. This is an area where we have green and red mixed very tightly together. And we're not really able to resolve this with a thresholding because the channels kind of like leak into each other, or it looks a little bit like leakage. It's not actually that, but you can kind of think of it intuitively as that. So you can see here, it doesn't actually, so even in the first channel, even if there's no cell here in this channel, it doesn't actually go all the way down to 0 from the intensity. It's more of a gray, and this is why we can't really get rid of it. But it's very interesting. And then aside from these areas where we have them very closely intertwined, we also have some areas which are kind of the borders between different areas of red or green cells. So in general, if you see this, and if you were a perfectionist, you might want to go back and increase both of the thresholds of your segmentation to get a little bit less of this leakage, to get a little bit less of this overlap. But for us, actually, it's very interesting to know that this exists. And this is, again, also why we always have to go back and we always have to check how did things work out. Here, for example, it's important information for us to know that there are quite a few cubes that are completely filled with green and with red from the segmentation. Now, the other thing that we can look at is the relative abundance. And that can be relative abundance of channel 1 or relative abundance of channel 2. And here we can see, if I put them next to each other, that they are more or less complementary. Of course, again, we have areas where they are both present, just like we saw before. That was this overlap. For example, this one here is like also present here. But in general, this is like, so this abundance, first of all, what does it show us? Again, it's between 0 and 100. So 100 is for 100% and 0 is for 0%. The 0 itself is actually black, which is why even though there are cubes in here which are part of the segmentation, they are completely black because there is no, the percentage of filling with this other color is 0, or the abundance of channel 2 is 0. So here we can see, again, we have this very distinct, usually the cubes, except for these areas where we can see there are a little bit at the border areas where they are not yellow or black. Most of the cubes are actually either 100% one color or 0% that color. And this is important to know. So now that we have done that, what is the next step in our agenda? Well, we want to see how these things develop over time, or how these things develop from inside to the outside of the biofilm. So what we have to do is we have to go to the parameter calculation and we have to calculate the distance to the biofilm center. And I'm just going to do that here. Luckily, I do not have to put any settings. I can just, right away click calculate object parameters and hope that it is done in a reasonable time. And this will basically give me my spatial resolution. And with the spatial resolution in this particular case of this particular colony type comes a time resolution. So again, the reason why this is taking a moment is because we have so many cells. And this is why I suggested for people who are following along and might not have a powerful computer to use the 2D version. The 2D version is faster because it only has one slice. And we just saw before that the slices are actually extended to 30 instead of 5. So this would be like 30 times faster. I guess maybe I should have done this too, but I wanted to show you a 3-dimensional example. Now what we will do is we will have a look again at the segmentation preview. So we do this like we did before by quickly switching to channel 2 and then back to channel 1. And then we just want to see how the distance to the center of the biofilm was calculated and if it makes sense. This is channel 2, now I'm going back to channel 1. All right, let's log the segmentation results. And then we can have a look at the distance to biofilm center. You can already see here that 2 different distances to biofilm center are calculated. One of them is the distance to biofilm center of mass. The other one, the distance to biofilm center at substrate, which is the distance to biofilm center of mass projected down onto the lowest set plane. And the reason for this is that in some cases, if you think of a biofilm as a semi-sphere, it makes sense to take the distance to the biofilm center as the distance to the biofilm center at the substrate, because then it's kind of like the center of your semi-sphere, if you had a whole sphere. Whereas in other cases like this one here, it makes more sense to look to about or think about the distance to biofilm center of mass, because we do not have this spherical radius. All right, so here I'm going to choose distance to biofilm center. I'm going to click on auto view, and we will see now what it looks like. You can see how fine actually our cubes are in this example, and you can see how it nicely, just like from inside to outside, changes. And this black area on the inside, this is just because anything below 200 micrometer or anything closer than 200 micrometer towards the center is black in this color map, and this is why just there's like this central area black. But there are actually cubes there. They're just so dark in here with a color map that we can't see them. All right. So now that we have seen this, let's have a look at the plotting. So now we have calculated everything we need, and the question is, how can we bring it into a plot? Well, first of all, we need to load the data as we always require to, and then we can think about what properties we want to plot against one another. And if you remember the plot from the presentation that I held earlier, you probably already know what the axes are that we're interested in. And those axes would be distance to the biofilm center versus the relative abundance. So what can we do? In order to do this, we want this 1.5D histogram again. We will use the distance to biofilm center as an x-axis, so select it, click on the plus. We want to make sure here that the range actually fits. So the range that is suggested that we can see sort of grayed out here is 0 to 40, which makes no sense at all for our colony, because our colony is gigantic. We can look again at this image here, and we can see that it goes up to 1,400 micrometer. So we want 1,500 maybe. So we uncheck this automatic range and instead put our own manual range. We choose 0 to 1,500. Another way could be, we could also keep the automatic range, but then go to this true range method and go from min to max. That would have the same effect. The bin size 50 should be fine because we have a lot of cubes along the way. And or maybe we'll keep it a little bit lower. And as a y-axis, we want to use the relative abundance. Let's use channel 1 here first. Let's see if everything's all right. All right, we want to change 0 to 100. We want the mean. This is correct. We want to scatter, which I guess also makes sense. do we want error bars? Well, in my opinion, it doesn't really make sense to do error bars, because what we want to know is has nothing to do with the variation. If you remember that we had either 100% or 0% of the cube being filled with cells fluorescent in channel 1, that would mean that the variance is also gigantic, because we're constantly going between 0 and 100. But what we really wanted to know is we wanted to know in this ring around the biofilm center, what is the average? Because that tells us something about the competition. So actually here, it doesn't really make sense to plot error bars. So we will not plot error bars, we can plot error bars, but they will be gigantic, as you can see. But well, let's keep them for now, or maybe plot it without them to make it a little bit neater. Maybe I was too fast. No, okay. So here we can see this abundance. We can see it starting actually below 50%, but then going up quickly. We see there is some kind of noisy part in here, which is probably due to these areas here where we have like whole chunks in one color. And then we can see it dropping down, which is due to what I mentioned before, is that we have kind of these flares going out in the other channel. So the other channel is going to go up in that area, because simply for very, very large distances, only these parts here, which are sticking out of the colony, are going to be in the plot, and those are all of channel 2. But I think that to get rid of this noisy part, actually, I would choose a few lower number of bins, so maybe 20. Just because it seems that otherwise the plot gets very noisy. And I think now I would say that this is kind of like smooth enough for my taste. Also, we started around 50%, which makes a lot of sense. And yeah, now we want to add to this plot the other relative abundance of channel 2. So we're going to select it. We're going to put it here with a plus. And then actually there is something down here that I haven't mentioned before. You can actually add a plot to the current figure. And when we do this, what happens is that the points get added in the same figure. And this is actually helpful in cases such as this one, where we want to directly compare. Okay, so now what do we see in here? We see a couple of things. We see that, for example, there is an area where it appears that they are somewhat, I forgot the word. Like as blue goes up, orange goes down. But then we can also see that there are areas where both blue and orange abundance is relatively high, and it is way higher than 100%. So if you add these 2 points here up, this is going to be way higher than 100%. Why? Because we have all of these overlap cubes. I don't have the plot right now, but you remember that we were looking at the overlap between the two channels in a cube, and we saw that there are quite a couple of cubes which have both in channel 1 and in channel 2 a complete filling. And that means that we are getting over 100% with the abundance here, which is fine. It's just we need to know why. And then we can see this one goes down. And as I predicted at this point here, the orange one is all the way at 90% in the end. And this is just because of this artifact that we introduced by not choosing the thresholding properly, because we wanted to do things fast. I mean, in reality, what you could do if you get this result and you know where it comes from, you could also say, okay, you cut it here. For example, you can say, I want to filter, or either I cut my graph or I filter here in the filtering gating option, and I don't take these cubes into account, which are so far from the biofilm center. Yes, but this is the type of plot that we wanted to get. This is the type of plot that we had in here, and I think let me bring it up. Qualitatively, it kind of looks similar. Of course, there are some variations just due to how I thresholded and everything. This is a little bit different every time I do it, I guess. Yeah, but this is the type of plot that you can get from this data, and this is how you get it. And we are now at 1/2 So I would like to check that Eric and can I see the participant list? I think I need to I can't see any of the Zoom controls anymore. Oh, now I can. Sorry.
We are already here.
You're already here. Perfect. Great. Then I think I would like to continue. And I think what I will do is I will quickly finish with the presentation that I have, telling you a little bit more about what is possible with BiofilmQ and how to continue using BiofilmQ if you plan on doing so. And after that, we will take a couple of minutes for you to have more questions, to give feedback, and to let us know how you think this could be improved. So I'm going to start here with the next slide. This next slide. And this is something that for those of you who have not been away in the last break, we already had touched on it very shortly, quickly. So here, the idea is that what happens if you want to use single cell segmentation? And this might be the case for some people. We know that a lot of people like to use single cell segmentation, especially when working with biofilms and confocal images, because they have single cell resolution and they want to make use of it. The idea is, and Niklas explained this very nicely before, BiofilmQ is not a state-of-the-art segmentation software, not at all. We only provide some very basic thresholding. We provide some very basic classical image analysis. And the reason why we do this is that we are not a lab that develops image analysis segmentation tools, basically. So we do not really develop advanced watershed algorithms. We do not, so we have some things that we apply to our data, but we know that there are other groups out there which are much better. There are amazing tools out there that can segment your data. There are amazing neural network architectures that you can use, and people are really putting a lot of effort into making them available also on this conference. I'm sure you have checked it out. Neural network, for example, learning, deep learning is becoming more and more popular and is taking over this field of segmentation with classical image analysis. So the idea of BiofilmQ is that you can either use this very simple approach of us, and you can threshold and then cube. And I will tell you that this is actually, for a lot of research questions, it's sufficient. This is why we provide the option, because very often it is sufficient. But if you feel like it's not sufficient for you, we have this option that you can import your pre-segmented data. And the way you do is you segment it with a different tool or with a neural network. And usually the outcome of that is going to be a label image such as this one here. And in this label image, every cell has a different intensity value. So the intensity value of each cell corresponds to the cell ID, a unique ID. So even if cells are touching, they will still have different IDs, and we will be able to tell them apart. And BiofilmQ can import this data, and it can turn it into a segmentation, and then the quantification is all done just as we had it before. So BiofilmQ is then basically a quantification and visualization tool. And as such, it's still very useful, I believe. So if you have pre-segmented data that you would like to use rather than the cubing method, you can do that too. The next thing I want to point out is just a few things that I have only briefly touched upon. So I have already shown you this custom parameter thing. I think I just wanted to bring it up again. That this is like a really powerful approach to define your own parameters if you find that BiofilmQ doesn't have the exact parameter that you need already implemented. We have these exports, and this is something that I've also shown you before, but here I want to show you an actual file that was generated by data that was exported with BiofilmQ. This was such a case where we imported pre-segmented data, then we exported it at some point to a CSV file after calculating some properties. And this is a rendering that you can find in PowerView. And this is like, you don't have to work forever to get this beautiful rendering. This is like, it's really easy, try it out. It's very cool. And here, for example, it is colored by local density. So this is a property that was calculated by BiofilmQ, then it was plugged into PowerView. So basically working with different tools to get this image. So the export is possible, batch processing is possible. I have talked about this, but we haven't really shown it to you in the BiofilmQ console. We have several options to do the batch processing. One of them is like a simple one where you basically just say, I want to do these tasks for all of the folders that are in my top folder, basically. You can also do it this way. This is like an advanced one. We can select the folders individually. And then this batch processing really allows you to run analysis, the same type of analysis on a whole bunch of folders without like constantly having to click buttons and wait, because I know how tedious that is, and I have done it so many times. The batch processing really makes your life easier. So this is also recommended if you're using BiofilmQ a lot, you start using it a lot, try it out, and let us know. Yeah, and if you have now started using BiofilmQ, if you want to keep going, if you need some more resources, I've mentioned basically all of these again, but I just want to have it on one slide. Where we have the paper that you can read to get some more information. It is going to be soon published in Nature Microbiology, probably at the end of this year or next year, early next year. Then we have the documentation online that I have shown you before, where you can find everything, including the video tutorials, which you, however, also find when you search for them on YouTube. There is this forum, and we are very happy to get in touch with you in the forum. People are writing requests also what they would like to improve about BiofilmQ if they have found any bugs. So this is really the place to contact us. If you have any issues, if you have very specific issues, you can also write us an email. But in principle, the forum is really a good place because you can search it. Maybe someone else has had the same problem. Maybe also someone else can help you. Currently, we're also always answering ourselves, but maybe at some point, if the community has grown, people can also help each other. And finally, a little bit of a shameless self-advertisement. If you just want to, in general, read a bit more into image analysis and specifically also about other tools out there, because I mentioned that there are other good tools for single cell segmentation, for example, out there. There are neural networks and everything. We have a summary review basically on the current state of the art, which is brand new from last week. And this might also be a good place to check out if you're just in general looking into image analysis, what is happening out there. And of course, the I2K is almost over, but I think we still have a couple of hours. And I will be around. I'm happy to talk to you. Maybe write me in the Slack first so that I actually go there, because I'm not constantly like, sitting there and waiting for people to come. But I am sitting there and waiting for people to come. And I would be happy to talk to you there if you have any other questions. Yeah, so just don't be shy. Ask us if you want to know anything. All right, and with this, I would like to also thank everyone that was involved. And I want to point out that these are a lot of people, like this list here, right? The list of names. This is all the people on the paper. So really, BiofilmQ has been a collaborative effort with tons of people, people who provided data, people who tested it, provided feedback, helped us make it better, helped us with the tutorials. And this is the group that Niklas, Eric, and I are working in. Knut is our PI. So everybody has really helped in this, and I'm very happy that we managed to get this tool out, and I hope that it will be useful to you.